Okay, so almost finished the last one on time. So this one's all about gene structure and regulation, so it flows on quite nicely. If we can get that slideshow open, there we go. So we sort of divide genes into those which are structural. So it's a gene that codes for RNA or protein product that creates something, that actually does something within your body, rather than regulate another protein. So it could have some sort of structural protein that we've talked about before, the enzymes, hormones, all those things that actually make you you. So they have a genotypic effect on your phenotype. We can see or measure what these are. We also have regulator genes, and now these are involved in controlling the expression of one or more other genes. So we've sort of got, there's some evidence here on my chin of some regulator genes that have basically switched off the genes that create hair colour as I've aged. It's a conservation thing. Why produce hair of colour when you don't need it? Of course, as I age, I apparently don't need it. It's got the hair, which is handy. So this leads us already to sort of this discussion about what a gene is, and there's a few really important components to a gene. There's a start instruction, a stop instruction. We've already talked about exons and introns, and this idea of a promoter region. These will all become very important. So all genes start with exactly the same instruction. Start. That is, start reading the information I contain and make an RNA molecule. And interestingly enough, in every single protein in all living things, they all start with methionine as an amino acid, because this is the one time it gets coded. So the code for start always codes this. Genes also have an instruction to stop, so stop reading and stop making the RNA. So the ATG on template DNA creates methionine and start, but the TAA, TAG and TGA, the triplets found in template DNA, all code for stop, but they don't add an amino acid. And that's an important distinction. So the initial RNA molecule, the pre-messenger RNA, includes amino acids for every triplet that's been read from the template strand. The final messenger RNA has removed the introns and kept the exons. So the exons code for a particular amino acid which will become an important integral part of the protein. The introns don't. However, there's been a little bit of work with a thing called reverse transcriptase, which allows you to take a protein and work back to messenger RNA and work back to the DNA and create the DNA it came from. However, having done that, if you take that bit of DNA and try and create a functional protein, it won't work. So there's something in the introns which is important to the function of the protein. And a lot of interesting stuff has been found in introns, I believe, including the breast cancer test, the BRCA1. Anyway, so introns, non-coding, exons, coding. They create DNA. They create proteins from DNA. Gene structure, there's also this idea of the promoter region. And it's just here, where's my thing? There it is. Just in this section here, before that first translation start codon. And so the RNA polymerase will connect to that. Oops, we'll connect to that first. I think I have to stop things. Have I? Not too sure. Are we still ticking? We're still ticking. Okay, sorry. I'm very good at this, aren't I? So this promoter region, which is where the RNA first attaches. I'll try and find there it is. First attaches to mosquito attack. To start the process of making the protein. There's a model for this which was discovered in prokaryotes, and I know Shania is planning on doing her outcome 3 experiment on this very process essentially. So the lac operon model was described in prokaryotic cells first, and it's now used as a model to explain gene regulation in all living things. So in this particular case, it's actually looking at E. coli, and it's the manner in which E. coli metabolize lactose when they have no glucose, or when glucose is in particularly low concentration. So we'll just go through this diagram and try and explain what's going on. So in the top diagram up here, this number one, this yellow here is our RNA polymerase, and it's unable to start reading the DNA at the promoter, the 3, because this green block here, 2, is in the way. Okay, so we've got a block in place, a regulator not allowing for the reading of this gene. So gene regulation. In the bottom diagram, these lactose molecules, and you find that here, these are all number 5s, bind to that repressor, the green block, releasing it from number 4, and allowing the RNA polymerase to start reading the promoter region and reading the gene. And we believe this is how all genes are regulated, this model. So, a lot of work currently being done in looking at eukaryotic genes and how they may be regulated, and this has opened up a whole area of really interesting research that of course might allow us to turn off and turn on genes which are causing problems for people. And I'm going to have to go get some air because I'm under attack all of a sudden. I'll finish this first. So, this actually belongs to an idea known as uniformitarianism. So it's an assumption that the same laws and processes that operate in the universe now, as we see it, have always operated. And so we're saying, if this operates in prokaryotes, it should also be the manner in which things operate in eukaryotes. And on the whole, it tends to pass muster. It was actually first described by a guy called James Hutton, a Scottish geologist, way back in the late 18th century, and finally it attacked a whole lot of ideas at the time, so it really didn't come into its own for another 50 or so years when Charles Lyell, another Scottish geologist, was able to find evidence for that in nature and finally made the case for this idea of uniformitarianism. And you'll see that in science all the time. Anyway, the other thing we're looking at here is a study of epigenetics. So how heritable changes, things we inherit, can be expressed or not expressed in the underlying DNA sequence. And there is some discussion of whether or not we pass on what are essentially epigenetic changes. Can I in fact stretch my neck and become a taller giraffe and pass it on to my offspring? So good old Lamarck may get a Guernsey just yet, but we'll talk more about that when we talk about genetics, evolution. And that's it for that little one. That's a shorty, isn't it?